Good morning, everyone. We're so excited to have our 2020 McNair Scholars Work in Progress Symposium. This is a long-standing tradition at the end of the summer before the new year begins to gather together and really just celebrate and take a moment to have all of our students together kind of sharing their research journey up to this point and the work that they've been doing over the summer. Obviously, since March, doing research has been even more challenging with the pandemic and access to labs and research sites and connections to faculty mentors. So I also just want to acknowledge just the amazing adaptability and creativity of each and every one of our McNair scholars in finding ways to maintain their research momentum or to pivot in a new direction so they can continue their work. And so this truly is a moment, I think, that I just want to invite all of our guests to just do a quick round of congratulations for all of our McNair scholars getting to this important milestone, which it really is. Obviously, research will continue in the fall, but this truly is a moment of celebration, and I hope that they can each just take a moment to breathe and take pride in the work that they've been doing this summer. So I also want to just welcome all of our guests that are joining us virtually this morning. We have family members and faculty mentors and campus partners and alum. So it's a great group. In the past, we've hosted this in the Dream Lab in the Night Library. But one of the things that is really a benefit and something we learned at the Undergraduate Research Symposium in May that was a bit unanticipated is this platform, this virtual platform, actually allows a much greater accessibility and engagement. And folks are able to join us and participate who normally wouldn't be able to physically come to Eugene, Oregon and to the campus. And so that part is really exciting. And so thank you to Keegan and Denise for coordinating this. And I think it's awesome that we're recording it because then it's available for other family members and friends and faculty mentors to watch asynchronously who also couldn't join us live this morning. So again, I don't want to take up too much time, but really just want to say how proud I am personally of where everybody is and how far they've made it through an exceedingly challenging year. So welcome, have a great time this morning. You're in good hands with Keegan piloting as the host. And so I'm going to turn it over to Keegan and his agenda. So welcome.
Wonderful. Yeah. So we have 10 scholars with us today. We have 10 presentations in a variety of disciplines. It has been a journey, I think, for Kevin and I to figure out how to help manage and advise all of these projects because they are in fields that Kevin and I are absolutely not in. But we have been very supportive and shepherding of a sort through deadlines and trying to connect you with people who can help you. And so over the past week and a half, I've been helping many of our students practice their presentations. I've been working with them all summer, hearing about all of the good updates and the challenging updates and the successes. And so all of these presentations, I think, are going to be very exciting. They might be a little dense at times. We have some very specific projects and some disciplines that I wouldn't necessarily automatically think of as accessible, but all of our students throughout the summer and focus on these presentations have really been all of them have been concerned about how to communicate about their projects and how to convey the ideas and the scope and what they're finding and all of that to people who are not in their field and to the people in their lives and to people in other fields. And so today will be a good exercise for them in that as you are communicating to a multidisciplinary space and to both an academic and not as academic audience. So this is, of course, part of the learning process. These presentations are works in progress in that many of you are still working on your projects. The summer is normally when our scholars conduct all of the research and they theoretically have a version of data by this point or by the start of fall term. Those classes will start next week. But for many of our students, due to COVID complications and other research complications, are still working on projects, which is completely fine and acceptable and understandable. Because if anything, this is better preparation for graduate study when you were trying to do research on a weird wonky timeline already. So this will be all the better preparation despite how stressful it may feel in the moment. So I promise, I know you all have been reflecting and learning things, so we will get underway as you continue your journeys in academia. So I will re-copy and paste the order of students. We don't quite have a strict timeline because there's like 13 or 14 minutes per student, and I didn't want to put a super like at 10:17, this student is gonna, I didn't want to do that. I didn't want to micromanage that much. So I will be keeping time as we go, but we will be going in the order that I just put into the chat. So students will be able to share their presentation getup as they are going up. And then theoretically, assuming all things go to plan, there should be time for a question or two or a comment or something from other people before we move on. But I'll be monitoring that, and I'll kind of pop back in after the presentation just to kind of make sure we still have time for everyone, because we have wonderfully 10 students to get through. So it will be absolutely delightful. If you have questions or comments during the presentations, please feel free to put them into the chat. And we can circle back to those afterwards, or the student can still read through those after their presentation and either private message with you or connect with you in some other way, shape, or form. Please absolutely feel free to connect with them and ask them questions about their research, because these students have been working on some super freaking cool projects. I've been so happy to be able to learn little bits and pieces with them this summer. So I cannot wait to finally show off our students to campus starting today. So it will be an exciting journey all around. I think that's everything, the last announcements that I have. And we are recording the session right now, and we'll be sending that out to everyone who RSVP'd. And we will probably have a link to it somewhere on our website once we refocus on that. So it will be an exciting journey available to everyone. So with that, I will ask or remind all of our students to briefly introduce themselves at the beginning of their presentation, but I will also put the program PDF document into the chat as well. And in there are bios and brief descriptions of all of our students and their projects and kind of where they're wanting to go with graduate studies. So please feel free to reference that as well as you go through. So with that, again, I apologize. I'm like, oh yeah, the program. I will turn it over to our first student, Emily Boyce.
Hello. I will mute. My apologies.
Hello. And I think Emily's mic is a little soft, so you may need to turn up your volume a little bit for this one.
My apologies for a soft mic. I will try and project. Hello, my name is Emily Boyce, and today I'll be talking about how plants deal with damage from light and how the plant ends up repairing itself. And this is the research that I have been working on not only for the summer, but for the past couple of years, actually. So we all know photosynthesis, the process that helps keep the biosphere in balance as it produces oxygen that we use and produces the sugars and starches we eat, as well as consuming an excess of CO2. We all know that this process is incredibly crucial for our survival. Now, to understand this process further, we have to understand more and more specific subunits of it. Let's start with the fact that photosynthesis isn't one process. It occurs in two different distinct reactions. The light reaction, which is dependent on sunlight, and the dark reaction, which is not. It is dependent on products from the light reaction. Both of these reactions occur in the center of a plant's universe, the chloroplast. Now, both of these reactions occur in different areas of the chloroplast along different parts of the organelle. The light reaction occurs along these thylakoid stacks along the thylakoid membranes where chlorophyll is generally found. This chlorophyll helps absorb the light that is later used. The dark reaction occurs in the stroma, which is the interspatial fluid, and is not bound by any chlorophyll requirements. Now, the chloroplast is actually quite an interesting thing. This organelle became a part of a cell through the process called endosymbiosis. What this means is that an ancestral prokaryotic cell to a chloroplast was actually engulfed by an ancestral eukaryotic cell and later integrated into the rest of the cell. This is also how mitochondria entered cells, and at least it's been hypothesized. Because of this origin, the chloroplast actually has its own genome. This genome now has to coordinate with the nuclear genome of the host cell. This coordination ends up creating the complexes that go through photosynthesis with different subunits from both genomes. Now, our lab cares about light damage, so we generally care about the light reaction. What this light reaction does is harnesses the energy from light and uses it to split water, producing oxygen as a byproduct, pump protons, and use those protons to power ATP synthesis that is later used to produce glucose. There are 4 distinct complexes in this pathway: photosystem 2, cytochrome B6F, photosystem 1, and ATP synthase. Each of these complexes have their own special structure in how they work and how they pass electrons. What's distinct about photosystems 1 and 2 is that they both have chlorophyll, so they're the ones that actually absorb light. Now, despite the necessity of light in photosynthesis, light is actually a damaging force, not unlike how skin cells absorb sunlight to synthesize vitamin D, but overexposure leads to a damage of cells. So how does a plant deal with this? Generally, there are 2 pathways a plant can go down, but both are employed. There are protective mechanisms a plant can employ and some repair cycling. Protective mechanisms generally include reducing the absorption of light you take in, or if you have excess absorbed light, reflecting it back off as heat or light. However, these are not always sufficient to protect a cell, and when that occurs, the cell must repair itself. Now, here's where the big question lies, because the repair cycle has been defined in very broad strokes, with light damaging a system, photosynthesis becoming inhibited, repairing damaged elements, and then you're back at full power. But we don't know a lot of the mechanisms behind this whole cycle. So that becomes a great question of research, and in each distinct complex, more questions arise, which is why our lab is focused on this research. Specifically, we care about photosystem 2 because it's the first complex in the pathway as it binds light. And in photosystem 2, there's a particularly interesting subunit that's very damaged by sunlight, and that's D1, as you can see right here. As you can see right here. What D1 does is it binds chlorophyll that actually splits the electrons. And then what else it does is makes up the core of the complex, making it crucial for structure. So despite how necessary this subunit is, it's very easily damaged by light, which makes it a very big target for repair, as it needs to be repaired very quickly. I apologize for the leaf blower outside. Now, how does the D1 repair itself? My apologies. D1 damage and repair is a very complicated cycle. It's only been defined in a few broad strokes, but those broad strokes at least are very clear, including the damage of D1, disassembly of photosystem 2, degradation of damaged D1, and synthesis of new D1. This whole complex, again, is really undefined in these intermediaries. However, what our lab cares about is synthesis, as that is the biggest part of this repair cycle. If you can't make more D1, the plant dies. And what's important about synthesis is that it involves the translation of psbA, which is the gene that codes for D1. Now, an important point to note for translation is that translation involves the translation of mRNA into protein. This is done through polysomes called ribosomes. And something very important for this is that the more ribosomes on a piece of mRNA, the higher the rate of initiation, meaning the more protein you're going to end up getting. This matters with psbA synthesis because psbA is a regulated gene, and it's regulated in quite an interesting way. It's regulated, at least in response to light. When a plant with normal psbA is exposed to light, it shows a very high rate of translation. However, when it's shifted to the dark, that rate of translation goes down significantly, as you can see. And then on subsequent reillumination, it increases significantly. We also know that psbA is regulated at translation, which is mRNA to protein, because of various ribosome and RNA experiments. Now, this is the only photosystem 2 mRNA that shows this significant a jump. Combining that significant of a jump and the fact that it codes for D1 makes it a very interesting point of focus for the repair cycle. And that is what our lab has focused on. Specifically, the question we're examining is, with all these observations of synthesis of psbA increasing in response to light and light damaging D1, how do those processes couple together to repair D1 in photosystem 2? Now, specifically, we're looking at the steps between initial damage and synthesis. So we're looking at kind of a big amount of intermediaries, but we're not focusing on disassembly or degradation. Now, in this process to figure out what's going on, a few factors have been looked at in the interest of seeing if they play a role. A few factors that have been of interest are factors that are actually seen in psbA reassembly. This has come with the precursor knowledge that reassembly is needed in the repair cycle, so assembly factors become of interest. A couple of protein factors that we've looked at are HCF136 and HCF173. HCF136 has been proposed to act as an inhibitor in the plant cell because mutants that lack this protein keep psbA translation very high, even in the dark, meaning it never goes down. The other protein mentioned here, HCF173, is also a known regulator of psbA mRNA as it binds to the mRNA, but it's also the only known regulator to do this, making it like a very important, or at least eye-catching factor. Now, there's also another factor. This is three proteins, actually, the HCF244 OHP1 OHP2 complex, or as I'll refer to it, HCF244 or the HCF244 complex. This is a complex made of these three distinct proteins that during assembly will bind proteins and pigments to photosystem 2. Now, it has been shown to play a role in damage in the repair cycle through ribosome experiments that show it lacking, but the role it plays is unclear. So we know it plays a role, we just don't know what that role is. It's been hypothesized that this could scavenge chlorophyll from damaged D1 or deliver chlorophyll to newly synthesized D1, but we don't know. With these few proteins and their hypothesized roles, and as well as a few key observations made in the past, there's now been a proposed model by Dr. Alice Barkin, my mentor, that has this complex as the hub of this cycle's mechanisms. So what our lab hypothesizes is that HCF244 complex coordinates with HCF173 to activate translation in response to damage. Future research questions will be focused on this communication between HCF244 and HCF173 and how these work together. One specific question that I'll be looking at is whether or not HCF173's footprint in a specific section of mRNA is necessary and sufficient to activate translation. Now, for a quick explanation for the model, because I understand this is quite a lot. This model posits a collaboration between the HCF244 complex and HCF173. What happens essentially is that HCF244 with damaged D1 will sense a need for newly synthesized D1 and communicate this to HCF173, which binds to mRNA and activates translation. Newly synthesized D1 inhibits HCF173, turning off that communication. When D1 is then damaged by light, that inhibition is stopped and HCF244 recommunicates to HCF173. So this is the model that we are looking at going forward and researching. Now the question comes, how do you research this? We use a few techniques for the bulk of the research, but specifically focusing on DNA, protein, and RNA and ribosomes. When looking at DNA for mutants or genotyping, our lab generally employs PCR to create a large amount of DNA that we can use for cloning or genotyping of our plants. We also utilize Western blots and antibody probes to look for proteins that are missing in specific complexes. Northern blots are also employed for RNA in the work that we also do, and ribosomal experiments are used to, such as RiboSeq, are used to quantify the amount of ribosomes on mRNA. At home, we do have some data, including literature reviewing and data analysis. However, for the work I've done over the summer, I have unfortunately, due to the circumstance of COVID, not been allowed in lab. So what I've worked on this summer is skill building and working on the background of our lab. Over the summer, I have learned some skills, including basic Python, to the point where I can make some pretty decent programs, including randomization programs and data analysis problems. I've also worked with my mentor on scientific writing and literature reviews to summarize the background of our lab's research to make it accessible. Lately, I've also had a chance to assist in the field in maintaining plants and propagating crosses. Going forward, my research will be aided by these because I have not only strengthened my background, but I have developed new skills that the lab can use. I'd now like to open the floor for questions and give special thanks to Dr. Barkin and McNair for all their support and help throughout this complicated, as one could call it, research cycle.
Wonderful. We have a moment or two if anyone has any questions or comments for Emily.
Hold on, sorry, I'm not seeing chat, my apologies. Ah.
Wonderful. Thank you so much, Emily. Or Barb, do you have a question? Barb, you're muted.
You want me to put in the chat or say it?
You can say it.
Okay, thanks, Emily. This is Barbara Jenkins. I'm the one of the librarians at the UVO Libraries, and my question is how does this situation that you're describing at the cellular level actually show in the plant in the field? Is there a specific kind of relationship between the way the mechanism that you're describing with D1 actually is demonstrated in the field by the plant? Can you tell, for example?
Yeah, there are a few ways depending on what's missing. So for example, if a plant has a problem with an HCF gene, they will glow red under UV light, and you can check for that because HCF stands for high chlorophyll fluorescence, so they give back light. It depends really on what specifically is wrong with the plant. Sometimes plants can show, like they can appear white or pale yellow-green if there's a huge problem with the chlorophyll. It really depends.
Okay, so
Barb, you're muted again.
you can't really tell very much at the physical level of the specific that's going on at the cellular level.
Yeah, I mean, we can get an idea, but it's really not specific.
Okay, thank you.
Wonderful. Thank you so much, Emily. Yay. Now we'll turn things over to Jyhreh.
Okay, give me 1 second as I share this. Okay, and we can start. Okay, hi everyone. My name is Jyhreh. I'm a senior at the UVO. And so today I'm actually going to be presenting my research, which is quantifying cranial shape change due to aging. So basically I'm trying to expand forensic aging techniques and understanding human ontogeny or growth and development through a geometric morphometric approach. And this is presented by me. And then also by my thesis advisor, which is Stephen Frost, because I will be using this as my honors college thesis and defending it later in the spring. Okay. So the inspiration for this was that I'm really intrigued by adult ontogeny. So what happens to us once we're adults and we're growing and developing. Also, I have a passion for forensic sciences. So if you can see in the little corner there, that is the logo for Bones. If you've ever watched the TV show, I want to be Temperance Brennan. That is going to be me. Also, I want to learn how to identify possible correlations between modern humans and also hominid growth patterns, which is basically our ancestors. And then also want to figure out what causes skeletal change as well. So some key things that I want to point out before I really get into the nitty gritty is the project known. So from a forensic anthropology standpoint, there are numerous methods of skeletal aging estimation. So this would be your pubic symphysis fusing, also your epiphyseal fusing, as well as like the ends of your bones or your epiphyses fusing together. That's also an identification method. Also primarily for the cranium. which is what I'm going to be looking at for my research. There's also two primary ways to look at aging in adults, and that would be suture fusion, which is like the little squiggly lines you'll see on a skull or later on in my presentation, and then also dental or tooth wear. And then from a human growth and development or ontogeny standpoint, the majority of what we know about human growth and development mainly focuses on prenatal to subadult, so like once you're developing in the womb, and then also once you reach the ages of 19 or 17. So project unknown. So this is where my research comes in. So are there any other cranial features that can be used to estimate age? And then also are age-related changes to the cranium, do they occur post-adulthood? So once you've reached adulthood until your death, basically. And then what do these changes tell us? So is it a genetic issue that's going on that we have to change as we age? Or is it due to wear and tear on our bodies as our skeleton does remodel itself each day? And as you get older, you notice that you mainly have arthritis sometimes or other things going on with your skeleton. So my objectives and hypothesis for my research is that the objective is to mainly identify cranial shape change that is correlating to age. And I have two key hypotheses, and then I have one null hypothesis as well. So my first hypothesis would be the human cranium does undergo shape change, fingers crossed. The second one would be changes will provide information on cranial growth and development. And then my null hypothesis is that there is no shape change. So we're hoping for hypothesis one, not my null hypothesis one. So keep that in mind through the rest of my presentation. So I'll just give you an outline of my methods. So we'll go through each one pretty quick, but I'm mainly going to focus on part two. So part one is photogrammetry, two is cranial landmarking, which is what I worked on this summer. So I learned how to landmark a skull, which was pretty interesting. Also generating a generalized Procrustes analysis, which is a superimposition where everything is in one space. And then also a regression analysis to show the rate of change due to age. Okay, so just diving in to my first method, which is photogrammetry. So this is actually creating the models themselves. So this image here, this is actually one of the models that was photographed. So these blue squares here is actually where the specimen was taken from different angles. So photogrammetry itself is the process of photographing a specimen from different perspectives in order to create a 3D or geometric image, which I'll show you later. So for my research, there are 50 specimens from the Terry collection at the Smithsonian Institute. I chose this collection because they are the only collection of specimens that are of known age. So I can know the person's age, as you can see here with specimen 34R. He is a male and he's age 44. You will be seeing this specimen throughout my whole presentation. So you'll go from my photogrammetry to my model making to my cranial landmarking. So you basically see what I do in the lab / at home every day. So I can take you into how I build my models. So they go from being photographed. So I have a top portion, which shows me all of the top features. So your eye orbits, and then also the top of your skull as well. Then I also have a bottom feature, which basically shows me where your mandible would be. Also underneath behind your ears, and then basically underneath your skull as well. So I go and I cut this portion to only show me the top. I cut this portion to only show me the bottom. This is what they look like when they're almost merged together. I just needed to make sure that they fit each time. And then this one is actually the complete merged skull from top to bottom. So I go from just having a top, just having a bottom, to actually having a full skull. So that was called geometric morphometrics. So that was me basically going in and making those photographs into 3D models. So I actually loaded them into Geomorph, which is a program that I learned to use in the lab before COVID kind of shut us down a little bit. But the point of geometric morphometrics, it's a method used to acquire and process and analyze landmarks. So the cool thing about using this is that it actually keeps and preserves the geometric relations among the specimens. So it still has the curves of an actual skull. It still has the surface of an actual skull as well, rather than kind of filling in the spaces. So this is actually what I did this summer. So this is cranial landmarking. So a cranial landmark is a precisely defined point on a specimen. So if you can think of a landmark as like a location drop pin. So say if I was here and I was like, hey, Keegan, like shoot me your location. I will have his drop pin and then I'll have mine to see the distance between the two. So that's basically what I am doing. You can actually see some here on this baboon specimen right here. So they have 26 and 27, and then those are the landmarks themselves. I'll have 34 landmarks as outlined by Langley et al. And I'll actually show you those landmarks in the next slide, which is pretty cool too. So I'm basically just graphing the distance between the two and using those measurements to assess change for each of the specimen. Okay, so this is actually one of my landmarked specimens. So if you can see these blue little dots here, so I gave you different views to show you all of the landmarks that are going to be placed on the skull. I know some of them are harder to see, but I can point them out to you. So each feature has its own little, blue mark on each side of the specimen, also on top and on the bottom. Okay, so it's important to note that these landmarks were chosen excluding the mandible, so your jaw, because it was not photographed during the photogrammetry stage of my research. Also, this list here, these are all of the landmarks that I used. So those blue dots that you saw on the previous slide, these are actually all of them. The little 2 indicates that they're on the right and left side of the cranium. And then it's also important for accuracy purposes that all the landmarks were placed in the exact same order, and this was done 5 times. So I have 5 times for all of my specimens that I have. So it's very tedious, but trust me, it paid off in the end, everyone. I loved it. So this is basically what I have left, which is creating a generalized Procrustes analysis, so superimposition, which is basically putting all the features in one space. So this image here, what I want is that I want to remove all of these nuisance variables, so like the position that it was photographed in, or the orientation, or any missing features that were on the skull. And then what is going to be left is shape. So this is actually the shape of a skull, and then if you can imagine it, that it would be the shape of a human skull once I do this portion of my analysis. And then also is just creating a regression analysis. So this is kind of what it'll look like. There'll be a straight bar here, age is going to be going this way, and then also degrees of change. So as the age increases, will the degree of change increase as well? So I just want to see, like, as we age, are we changing? Are we not changing? Hopefully we are changing. There is another study that is done in my lab that used actual primate skulls, and she saw a degree of change. So we're kind of working together to figure that out as well. But yeah, I want to say thanks, everyone. You've all been a huge support system. Trust me, it takes a village, and I know now that it really takes a village. So thank you, everyone, especially McNair. Daphne Gallagher, I saw that you were in here as well. And my mom. Hi, Mom. I love you so much. Thanks for answering my phone calls and listening to me, even though sometimes you don't know what I'm saying. But yeah. Oh, hi, Christabel. I didn't see you in there either. Hi.
Are there any questions or comments for Jyhreh really quick?
Oh my, I'm sorry, but I have to say, Jyhreh, I'm so proud of you. And I'm glad that you believed in yourself. You see, I have it. Thank you for believing in yourself. All the way from Louisiana, we love you. We love you. I'm going to send you some gumbo, girl. You did a good job.
Thanks, Mom.
I have one, Jyhreh. This is Barbara Jenkins. So once you've done all these things, what does it tell you about, so can you, let's say you find that there is change in the human skull, based on your research, literature review and so forth, what does that lead you to think about in terms of, if there is change, where does that take us in terms of thinking about our skulls differently?
Oh, I love this question. Okay, so as I mentioned earlier, we normally stop at like age 19, so in our sub-adulthood. So that lets us see that we're still changing as we reach like into like our later stages in adulthood. And then also it can help us see if there's also correlations between hominid growth patterns as well. So our earlier ancestors, to see if we had the same or if they had the same kind of skull shape change that goes on, which actually bridges the gap more between what we know and what we don't know, which is why I was super interested in doing this, because I was like, okay, no one else is kind of doing this with humans. We've done it with primates. So let's see if we can see the same thing with humans as well. Also from a forensic anthropology standpoint, you can have like a better age assessment or age estimation for you can say individuals that you find at crime scenes, because that's one of like our most things. So I've learned in forensic anthropology that you have like the big four that you look at, which is age is one of them. So that can help us better estimate age of those victims, and then also kind of put the subject pool, the victim pool to be smaller in solving those cold cases or those cases that are already open.
Okay. I have a follow-up question, but I'll lend the floor to someone else if they have a question.
I do have a question. Does this research, like, how does your research contend with the two different sexes? Are you just kind of like, how does it deal with the two sexes? Is it different, or are you treating it as the same?
Yeah, so of my 50 models that I have, 25 are male and 25 are female. So in the kind of Procrustes analysis, that will be kind of like a nuanced variable, so it'll be taken out. But for further research, it can be, if I wanted to keep male or female, if I wanted to keep sex in there, I could just take out another variable and say, this is what happens in males, this is what happens in females, and also try to find the difference between those, if there is one as well. So it can be done. It's just, that's just a further implication of what I could do, say when I go to grad school, or if someone else wants to pick up where I left off with doing just age.
Oh, cool.
I saw quickly that there was a question from Daphne in the chat. When you measure your landmarks, are you measuring them as a straight line or across the surface of the bone? And why?
So when I'm landmarking, there's an actual option for you to do curves. So it actually keeps the shape of the skull. So when I do my superimposition, I don't get the jumbled mess that you saw in the first one. I actually get the outline. So sometimes I do curves, or other times I do patches, where if the landmarks are so close together that it can count each one rather than having them go kind of like straight and straight. But when I do, say if I do from this side of your head to this side of your head, because you have two features there, I do make sure that they are in a straight line to make sure that one's not too high and one's not too low. So it has the proper distance.
Wonderful. Thank you so much, Jyhreh. Yay. If you have other questions, please feel free to put them in the chat, but I do want to make sure that we stay on time as best as we can with things. So I'm going to turn things over to Lukas now.
Thank you, Keegan. Great job, Jyhreh, and also to Emily. The morning is going strong so far. Even with leaf blowers and lawnmowers, because I have a lawnmower, so that might come in, just for like forewarning. But yeah. So, hello everybody. My name is Lukas MacMillen, and I'm an undergraduate researcher with the Sing Lab in the university's Institute of Ecology and Evolution. And today I'm going to be talking to you about condition-dependent recombination and the literature review that we've been working on over the course of the summer. So beginning with phenotypic plasticity, we know that this one genotype, multiple phenotype response is pervasive in nature. We see this in dung beetles, where a horned or non-horned phenotype is dictated by larval nutrition. The pattern intensity and organization on these butterfly wings are determined based off of the rearing temperatures that they're raised in. And the general mass of what is called the water pepper smartweed varies with the percentage of daily sunlight exposure. And before going on, I wanted to just emphasize that all of these sort of morphological variances are stemming from the same genetic background. And the thing that causes a difference in morphology is this variation within the environment. And that is what we call this one genotype, multiple phenotype response, depending upon a cross-environmental change. And we see this in recombination, which is the reciprocal exchange of genetic information between two homologous chromosomes. And we've seen this for more than a century, where meiotic recombination, or recombination that occurs during sex cell production, sperm or egg, changes with respect to environmental cues. And I wanted to note, as it shows down here, that all of these cues that I've shown are from Drosophila melanogaster, or fruit flies. And this graphic below is just to kind of show a trend where we see, as the environment changes, or temperature across the x-axis where it's increasing, recombination rate changes over time. And this is recombination rate increasing across the y. And so you can see that as temperature changes, there is a difference in recombination rate amongst flies. And so then the question becomes, like, what are the evolutionary consequences of recombination for populations in nature? And I wanted to take us to a sort of simplified model of like how recombination might operate within an organism in terms of for future generations, where we consider like these two strands presented below, where each icon is represented as an individual allele or location along a chromosome. And now you can imagine that you're a huge fan of the Apple Messenger app and you can't stand like the associated operating system, but likewise, the lack of blue text messages that are featured in the Samsung is just like really bothering you. But you really like the fact that Samsung allows you to personalize your home screen because Apple doesn't know how to do that yet. Well, one benefit of recombination is this idea that it can bring together the favorable allelic combinations. Thus, you can have that dream text operating system setup that you've always wished for. But then a question comes kind of, what happens if you already had the ideal setup? Say you prefer the Apple OS and Messenger. Doesn't that mean that recombination can also break apart beneficial combinations? And indeed it does. That's where we see that recombination can both bring together beneficial alleles or it can break them apart. And this nature of recombination suggests that the process would be tightly regulated, but empirical studies like the ones that I showed previously have supported otherwise for the past century. And this is that idea of them being plastic, because you wouldn't expect something to be both tightly regulated and readily plastic. Thus, research has been left to question how plastic recombination has evolved. And one theory on the evolution of plastic recombination is supported through the lens of organismal fitness or condition. And what organismal fitness is, at a glance, it's this measure of how capable an individual is at surviving to a reproductive age, and then reproducing, and then also how well the genome of that organism can be retained throughout generations. And the fitness-associated recombination, or FAR model, predicts a negative correlation between organismal fitness and recombination rate. This is shown in this figure below, with fitness increasing across the x-axis and recombination rate increasing along the y. And now this red circle showing one extreme is where a condition is poor, so the organism has low fitness, and recombination rate is expected to be high by this model's predictions. And this expectation is due to the fact that the chance benefits of recombinations would outweigh the chance costs. The benefits being bringing together new combinations of favorable genetic material, and the costs being breaking apart these favorable combinations. In a low fitness state, the likelihood of highly favorable combinations being broken apart is less likely because your overall genetic fitness is lower. However, for this green circle, showing the other extreme, where condition is good or higher than the previous, we see that there's a lower recombination rate due to the fact that these chance costs now outweigh the likelihood of any chance benefits occurring through recombination. With this, our lab asks two guiding questions related to the FAR model and the condition-dependent recombination hypothesis. And those are, what evidence supports this model's expectations, and how have empirical studies investigated the effects of fitness on recombination thus far? We've approached this question through the literature review process, where we had bi-weekly meetings over the course of the summer to discuss assigned literature. And I, as an individual, had meetings with my PI, Nadia Singh, to discuss section-specific literature, and then the drafting process once that literature was finished. Thus far, we've read 45+ pieces of literature, and we're currently working through theory papers, and we are also working through the sections specific, the drafting process of the sections. That being said, my summer was dedicated towards this section on condition-dependent recombination in Drosophila melanogaster, a fruit fly, and we're now going to delve into some of the empirical findings that drive my section of the review. And beforehand, I wanted to show a brief overview of the assortment of what the data sets kind of look like related to the farm model. But I wanted to note that we're only going to be going into and discussing the ones that are like sort of more digestible, or more like the trend is more similar to that as that like simplified graphic that the farm model wants to predict. So to begin, this paper from 1981, the researchers collected flies from Berkeley, California, and they have 70 unique wild type genetic backgrounds. And then they investigated the effect of this genetic background on recombination rate and organismal fitness as independent variables. And then they summed their findings in this figure, which shows the correlation between the two of them. That being said, fitness is shown increasing across the x-axis, and recombination rate is shown increasing across the y. And we're now going to go through the findings of each of these fitness assays used, where these three measures are different fitness assays that they use to quantify organismal fitness. Looking at this, we're looking at viability, and I'm going to go into it in 1 second, though I wanted to note that in my sort of recreation of this graph, I placed these dark triangles at the locations that they had. You can see that here. And what these represent are fitness classes, which are determined for each fitness measurement. And it's sort of just like a home base to place these 70 genetic backgrounds. So that way you can kind of see a trend line across varying fitness patterns. Going into viability, what this is you collect fitness data by comparing the proportion of progeny produced to that expected by a value given by Mendelian genetics. And notably, this trend line represents a correlation non-concordant with the farm model's expectations. So it doesn't show that negative correlation between fitness and recombination rate. Male fertility, on the other hand, which measures the success rate of males having like copulence or sex by like visualizing how much sperm persists after some sort of sexual event, we see that there is a significant correlation concordant with the farm model's expectations, where this negative correlation is like present. Likewise, in female fecundity measures of fitness, which counts the total number of eggs produced by a single female after some sort of bout of sex or copulence, it found also a significant correlation concordant with the farm model's expectations. Thus, the data from the male fertility and female fecundity assays show the negative correlation expected by the farm model. And the main takeaway from this figure is the idea that the genetic background affects both organismal fitness and recombination rate in a manner that is concordant with farm model expectations, at least between two of these three assays used. We're now going to take a different shift and look at a paper that deals with heat shock exposure on recombination rate. And I wanted to note that heat shock exposure is, there's a lot of jargony terms that I'm using throughout this, and I would suggest that you don't need to necessarily remember exactly what heat shock exposure is, though it is this experience of some sort of very high heat event present on an organism. And that's being measured on the x-axis through increasing time intervals of this heat shock exposure event. And we're just going to be looking at how this, as an environmental factor, interacts with both organismal fitness and recombination rate. So that being said, recombination rate here is shown increasing across the y-axis. And what we see is that recombination rate is significantly increasing as this heat shock exposure time also increases. Additionally, this paper found an effect of heat shock exposure time on organismal fitness, where fitness is shown decreasing across the y-axis. And here, I wanted to note that organismal fitness is labeled as the number of offspring, and this takes into account two things: that idea of how many offspring does a female produce, and then also how many of those offspring can live to then produce future generations on their own. And this is that idea of how fit is an organism to be able to carry on its genetic material throughout different generations. So putting these two figures together, what we see is just like data that is also concordant with the farm model's expectations, where recombination rate is increasing across this bout of across heat exposure time, whereas fitness is decreasing across this heat exposure time. And that brings us to sort of a summary of the general findings from literature thus far, or the papers related to my section. The first one is from this previous slide, the first slide, I suppose, where organismal fitness and recombination rate tend to vary across wild-type genetic backgrounds. And I wanted to note, as you can see in bold, that another paper in 1986 found very similar results, but they also use very similar methodology, and one of the authors was the same. Additionally, one that we didn't go into, but shows that work with deleterious mutations has an effect on fitness and recombination rate, concordant with the FAR model. And lastly, that idea of heat shock exposure being shown to produce FAR model expectations through that negative correlation. And just from this slide alone, you might be asking yourself, did I only read 4 papers this entire summer? And no, as mentioned previously, there were like 45 papers that have been read and discussed, and the process is still ongoing. However, I wanted to, which is shown here, this just like the sort of populated folder that we had for my section specifically at the beginning of summer. And what you might note is that only these bottom 4 papers, the ones with like the red circles, were the ones that had data that was actually significant for the condition-dependent recombination hypothesis. And then you might be questioning kind of what is lacking in these papers. And to start, it's this idea that fitness analysis has not been standardized, and many of these 6 papers lack fitness analysis at all. And what that means is that it's not that they failed to show an effect of fitness on recombination, because that'd be very useful information. It's that fitness was either assumed to be impacted and therefore not tested at all. And so they only measured recombination effects and then didn't have a correlation between recombination and fitness. And this brings us to sort of the significance of both the review and of the FAR model and the evolution of recombination in general. Specifically, future research to figure out the FAR model and its validity needs to focus on like standardizing these fitness assays, which is kind of for reasons that I previously mentioned, just to increase the effectiveness and validity of data collection. Because if we don't have know what methods work to yield significant data sets, we're just going to kind of be like twiddling our thumbs for decades and not really finding enough information. Along with this, there also needs to have this like redefinition of how we label stressors, like environmental stressors, because as it stands, environmental cues are far too often labeled as being stressful. And in this instance, if an environmental cue is labeled as being stressful, some of these researchers, which this also came up in those 6 studies, then assumed that fitness was being impacted, because if an environmental cue is acting stressful, is acting as a stressor on an organism, fitness is also potentially being impacted. And so that's why the research world must take care to find out these like bioindicators of stress, something that we can look at in an organism and then say, oh, okay, well, this is now standardized and it seems like it's varying. So maybe there is some sort of stress impact on the organism. And then we also want to be able to support those findings through these standardized fitness assays. And with both of those at our disposal, the evolution of plastic recombination can be like further investigated. And this includes, but it's like not limited to, the model of condition-dependent recombination, because it's important sort of as we close that condition-dependent recombination is not the only model of the evolution of plastic recombination. And so that wraps up my presentation about my summer research. And I wanted to give acknowledgements to every member of the Sing Lab who's been super helpful over the course of like reading these papers and working on the review, my PI specifically for meeting with me individually and giving me the time that they did, and also all of the McNair staff who have just been very supportive and helpful over the course of since winter. So that's almost, I think, 9 months. But yeah, thank you all.
Yay, thank you so much, Lukas. Yay. In the interest of time, wonderfully, I might move us to the next presentation. But if you have questions or comments for Lukas, I want to please encourage you to put them into the chat. I just want to make sure we don't accidentally take time away from the students we still have to come. But as you can see, these are super cool and very complex projects. So again, I want to reiterate my admiration and respect for students trying to put all of this information into these tiny little presentations that we have forced them to do. So with that, I will turn things over to Kat.
Hi, great job, Lukas. That was awesome. Okay, so let me go ahead and share my screen here. So my name is Kat Luna. I hope you guys can see all of that. I am an undergraduate in the psychology program. And I just wanted to have my kids interrupt me for a second just to give you guys an idea about a little bit of my research. So I don't know if you guys could actually hear them or not, but my research is on how non-traditional student parents deal with their daily stress and time management during COVID-19. So, all right, guys, you're good. So that was staged, but that is a daily occurrence in a lot of non-traditional student parents' lives. So I would love to thank all the McNair cohort that I have. You guys are all amazing. And also to Keegan and to Kevin for their ongoing support. And Barb, you're also great support too, just Trio in general and my whole like non-traditional student community. So, all right, here we go. So my research question is, do non-traditional students with children experience higher levels of daily stress when managing a weekly schedule compared to other students? And this may seem a little bit obvious, but my research is leading me to wonder if that's actually true or not. So what is a non-traditional student? Who are they? These are students who have taken a non-traditional or non-linear path to pursue their education. So folks that are a little bit older, maybe they may have taken an extended break from returning to school. They may be student veterans, or they may just self-identify as non-traditional. And also non-traditional students could be students with children. So anyone who is caring for children while pursuing their education is considered a non-traditional student parent. So how do students manage their daily schedule during this time? And this looks different for any particular student. So some, might keep a calendar available. Some might use digital reminders on their phone. Some students might just check their email often and have an incredible capacity for remembering tasks. For this study, I'm developing a survey on how students' perception of their ability to manage a schedule, and we'll be using self-report data to compare these results. And so one of the tricky things about this study is how to operationalize daily stress. And because daily tasks take different forms for different folks, each self-report will be based on the perception of how stressful these daily events are for each student. And so this is something that I'm still developing right now, and we'll be using a well-being focus for measuring daily stress. So why should anybody care about this research? One of the big reasons is that during COVID-19, everybody's well-being, it seems like, has been impacted in some way. These folks, the non-traditional student parents, have a limited flexibility of schedules compared to some other students. And so, because of all the school closures happening and they're limited on being able to be available during the day for their coursework. So on a study from the National Center of Education Statistics, it says that universities that participate in federal aid programs are required by the government to report information on traditional students only. So that means that, anybody that's under the age of 24 and pursuing a degree for the first time, their demographic information is tracked by universities. But that means that we don't have reliable data on 65% of college students, and that includes student parents. So that leads to an underrepresentation on campus for these folks and their needs. So a limitation of resources on campus could be a result of that. And one of the limitations and resources is childcare. And childcare is a really important aspect for student parents who, you know, have a lot of interruptions, as you guys saw, and are managing, you know, their schedule and their child's schedule. And so a lot of folks who have their children pulled out of school and, you know, are becoming essentially their children's primary or secondary educators are, you know, lacking in being able to have an environment where they can actually study and an environment for their children to socialize. Another impact this could have negatively on student parents is in role tension. And what I mean by that is being able to kind of switch and shift in and out of being a student, being a parent, being a partner, being back to a student. And so a lot of parents, you know, can relate in that it's really hard to switch, you know, on and off and just kind of jump between the two and get right back into your focus. So some of these impacts could, you know, lead to lower retention rates in students kind of dropping out before their program is through, to be able to maintain a certain level of support for their children and their families. There's also a negative impact through traditional gender roles. So a lot of women feel a little bit more of this impact negatively if they're in the home and they're their primary caregiver and they're kind of maintaining this role as a mother to be able to care for their children. There are also significantly more barriers for single parents going through their degree because they are, you know, they're the only ones in the home, so they're the ones providing all of the support for their children and also maintaining their own goals and, you know, accomplishing those daily tasks. And then there's also lower rates. Studies have been done showing lower rates of graduating on time for these students, not only non-traditional students, but non-traditional student parents specifically. So that's 6 years that it might take for students to complete their degree could be drawn out a lot more with parents having to work and support their children while going to school as well. And this, I didn't put in the slide, but also lower GPAs as well. Just the focus in general is taken away from their studies and placed onto their children's needs. So the significant impacts of why this study is important, and I just want to make sure to kind of drill that. And so in previous research, Predictors of Well-Being in Female College Students with Children 2006 study evaluated predictors of well-being in female college students with children. So participants in the study reported challenges of balancing personal life and relationships with academic tasks, emphasizing the demanding aspects of these factors as having a potential negative effect on well-being. So results of the study showed that higher levels of social support and internal structures of self-confidence were predictors of academic success. This study contributes to the idea that both external and internal factors play a part in student-parent academic success, with a higher likelihood of negative external factors contributing to the distress of the individual. So this is why it's so important that, you know, before school closures, that these parents had this external support within their community and their environment. And this is something that has been impacted severely through COVID-19, where they don't have as much social support as they used to, and external support. So in another research in 2019, a study that was conducted by the Hope Center for College, Community, and Justice, over 23,000 student parents responded to the Real College Survey. And based on their survey, more than 1 in 5 college students is a student parent. So student parents reported experiencing basic needs and securities during their college experience. And among these basic needs were childcare and then various campus social support resources. And I put a little picture up here that says, if you knock on this door in the next 80 minutes, you'll be cursed to poop your pants forever. And this is just one silly technique that has been used by a student parent in their undergrad program to be able to maintain their environment during a final. So that way they have some space and some privacy to be able to complete their coursework. So So my hypothesis is that non-traditional students with children do experience higher levels of daily stress compared to traditional students while managing a weekly schedule during COVID-19. And that may seem a little obvious maybe, but non-traditional students often are capable of managing daily tasks like work and personal life before they decide to return to pursue an education. And student parents have even more demands on their time and develop the ability to juggle and multitask, you know, and optimize their time during the day. So it's possible that managing a weekly schedule could decrease their levels of stress as a result of this pandemic. So my methodology is I'll be using convenient sampling of undergraduate students for simplicity purposes to demonstrate the knowledge of methodology. A questionnaire including basic demographic information will be given to students to differentiate between groups and determine if they're non-traditional versus traditional or if they have children. Simple procedural methods using Qualtrics survey software will be used. All participants will be emailed the survey and asked to complete it digitally, so adhering to social distancing policies. Survey questions will be designed to include psychological distress, self-esteem and satisfaction with life measures for well-being, and overall perception of time management abilities, along with self-efficacy expectations. All participants' information will be protected and any data reported will be anonymous, complying with the Institutional Review Board or the IRB ethical standards. An analysis of variance or ANOVA will be used to analyze the differences among these groups. And then a statistical t-test will be used to compare the two groups and test the null hypothesis. And so the results of this, we don't know yet. I'm really excited to find out. The results of the study will help us see if there's statistically significant differences in mean perception of daily stress using weekly time management techniques in undergraduate students compared to non-traditional students with children. The limitations of this study is that I'm using convenient sampling. It's not representative to the general population. So future studies could use random sampling to offer generalizability. Limited sampling from this location here at UO that could prevent cross-cultural analysis that could expand and see if there's actual differences between cultures. Sampling and comparing data during a pandemic definitely isn't representative of the normal college experience. So that's a big limitation as well. So non-traditional parenting students find themselves faced with even more challenges than the average student and often depend on organization to carry them throughout their week. And more than ever, parenting students are experiencing unprecedented levels of daily stress and need resources on university campuses to help them become successful students. And with the changing environment, policies restricting in-person education opportunities brought on by COVID-19, parents with school-age children find themselves becoming their children's first and secondary educators due to these school closures. In addition, they're becoming their children's go-to source of socialization. So these changes in already limited college experience for students with children pose even more challenges and risks associated with degree completion and may lead to lower retention rates, as discussed before. Many universities are unaware of the increasing burden that this places on the parenting student and their ability to cognitively shift their focus towards each responsibility throughout the day and over the course of each week. The college experience is filled with diversity, and the less equitable the college experience is among our student populations, the less enriched we are as a community overall. So this research could lead to creating better policy around accommodations for parenting students who are experiencing conflicts with schedules, focus, parental responsibilities that might prevent them from attending synchronous courses during the pandemic. Future research might also focus on the new normal after the wake of the pandemic settles a bit, hopefully, and work towards finding differences among student populations that are pursuing degrees and ways to help them access better resources to assist them in being successful. So, and that's my presentation, guys. I hope that if you get a chance to see a non-traditional student on campus or have them in your classes, reach out to them, encourage them. Where I say we, because I'm non-traditional, are incredibly resilient, and this experience has been so rewarding to me to be able to balance that home life and my family and be able to pursue these huge goals in life and be able to have a generational impact on my children. And hopefully in the future, these, kind of opportunities for equity among the university populations will help be able to create a broader and more diverse environment for all of us. So yeah, that's my presentation. Thank you. Any questions?
Hey,
Maybe there's some
Kat.
stuff in the chat. Let me look. Thank you.
Amazing, amazing. I might move us
I
to the
can't
next
even
presentation.
scroll through, so okay, sounds good.
Yeah, no. But if you have questions or comments, please absolutely put them into the chat. There's still been some engagement about some previous presentations in there, so please feel free to use that. But in the interest of time, because I love you all, but like we need to get to other people. I swear, I love all my children. I'm gonna move us now to Alexie.
Hi everyone. So I know we're in short manner of time, but I just think everybody needs to see this really quickly. I'm never left alone. I don't have real kids, Kat, but I feel the distraction element. I definitely do. Okay, so share screen. Dun, 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 dun. Share. Okay. So, present. All right. So I have a very overarching research topic, as there's a lot of things that I'm choosing to include in it, and I will explain in great detail throughout the paper and generally in this presentation how all of those things are going to connect. I'm doing my research with Michael Maikov, and he's been super awesome this whole time. However, he's been back and forth between Russia and here, so I've been doing a lot of finding things on my own, but independent queen, we got it, so it's not a big deal. But yeah, so let's see here. All right, so the overview of my project that I've put pretty generally is just I'm interested in connecting the concepts of human cognitive behavioral analysis, inescapable human biases, and how they are utilized to understand how game theory hypothesizes the way politics and the two-party system works now and how it will behave in the future. Obviously, that's pretty general, like I said, but I'm going to get deeper into how all this is going to work. So here's just a couple pictures. On the left, I have an example of the Prisoner's Dilemma game, which I'm going to talk about a little bit more later. The top right picture, I think, is just an interesting idea, imagery of psychology, of going with rationality in the brain or emotion in the heart. And then the bottom right picture is a diagram associated with cognitive behavior analysis. So I have been working on a lot of research this summer between different articles, between different books, between just all sorts of scholarly work. I broke down what the Prisoner's Dilemma game is, and basically it's just a game that's created by a police officer because he had a suspicion that the two criminals that were brought in under one charge may have also been culprits in a separate robbery. And so this game/model is a paradox decision analysis where two individuals act in their own self-interest and do not generate the optimal outcome due to lack of knowledge of what the other player is going to do, and therefore a lack of trust. So this shows us how human nature leads us to choose defection over cooperation because it's more rational and the self-benefiting choice, which basically just goes to show that in general, without repeated occurrences of things or an analysis of the repeated occurrences of things, people are going to do what's best for them, and that correlates a lot of the time into politics as well, which is why there's a lot of corruption, especially in a society that is based off money. And I think that it's really important to be able to pinpoint where these deviations happen so that we can prevent them in the future, or at least be aware of them, so that people that are voting, that are actively participating in our government, are much more aware of the choices they're actually making. So the Nash equilibrium is directly correlated to the Prisoner's Dilemma game, and it's the optimal outcome that I was just talking about earlier. And basically it's just talks about how there's no incentive for the people playing to change what their initial strategy was, even after knowing what the other player's choice is. And so that just goes to show that after a couple games of playing the same time, they know what the other person's going to do. And it's just a model to show that people in human nature and within their biases are not so easily pushed away from what they choose to be emotionally attached to. And so I also am going to go deeper into that about how multiple repeated games actually moves people to choose cooperation instead of defection, and that is based off of mathematical calculation as well. But that comes in way later, so don't worry about it right now. Another thing I've dived into is tribalism, which is more of the psychology side that I said I was bringing into this. And so basically I've been doing cognitive behavioral analysis, which has led to the necessity of understanding human nature and evolution, or lack thereof. And that, I mean, basically we've evolved so quickly in so many ways, like with technology and everything advancing so quickly, that we also are incapable of moving away from innate human tendencies, which have to do with, you know, life and emotion and things like that. So it's very interesting to look at tribalism and how we always associate ourselves with people that we feel belong in our tribe, that we think the same as we do, that we don't really argue with, because then we put those people on a pedestal and then treat other people differently because they aren't associated with our tribe. And that feeds deeply into politics as well. As we can see, there's a lot of diverse opinions on people depending on what party you associate with. And then getting a little bit deeper into that and a little bit worse is infrahumanization, which is another thing that I actually hadn't known about before I started this project. So it was really exciting to do some more research about that, and I'm going to be progressing my research on this topic as well. But basically, as the definition goes, it's that you consider anyone who's an outgroup of your in-group completely dehumanized. And that affects everything from the way, you know, politics goes, the way people discuss laws, the way people treat each other, even just based on race, gender, sexuality, all sorts of things like that. And that's become a really huge, obviously, problem. And it seems to be a growing problem since the current leadership we have has been really pushing those things out into the open, I think. So the connection between all of these things that I have been working on showing, and the reason why I'm doing my research, is basically that politics in general and game theory always pulls from many different areas of education, and it requires, like, I remember Michael, he always talks about how if you're going to be someone who's into game theory, you got to be able to pull from a little bit of everything. You got to know economics, you got to go math, you got to know psychology, you got to know politics. Like, you have to know a little bit, or not a little bit, but enough of all of it in order to make accurate predictions and know exactly what it is that you're looking into. And I think that's why I really like game theory, because I feel like human nature and politics is so diverse for these reasons, because it really does pull from all of these aspects. And I think that because they are all connected to human behavior, human evolution, human emotion, politics, that it's really important to understand all of these things in order to fix problems now, fix problems in the future, and really understand the two-party system and how we can overcome these differences that really shouldn't be counted into importance of why we choose to not agree with one another. So just expanding onto that part, just making it a little bit more concise. The first point is I just want to be able to better understand human behavior when it comes to politics and evaluate how we can overcome issues in the future. Second point is to diminish tools and thought practices that separate people and ideology associated with inferiorities, based on gender, religion, sexual orientation, race, and etc. And then the third point of why I want to do this study is to prove how deeply intertwined emotion and politics truly are, especially because a lot of people try to claim that they are super rational, which is okay, but we often can't escape our emotion, especially when we're making decisions. So the methodology I have been working on this summer is that I've been collecting data primarily in scholarship work. Some of it's derived from books. I've been avoiding the experimental data processes. I am going to be backing my paper up with facts, and I'm going to do my own experimental work, which is also why I've talked about sampling data in the next bullet point. I just need to figure out with Michael the next time I talk with him exactly what we're going to do, and I also need to talk with Kevin at some point, so that's going to be happening very soon. The data collection that I'm focusing on is the sampling aspect, which will be pretty much like surveys or focus groups, obviously because of COVID-19. Not entirely sure what's going to happen with that yet, but we'll get there eventually within the next few weeks. I'm not super worried about it. And then the qualitative data is going to be the most important for my data analysis because I am doing work that is exploratory. So my fall term goals and objectives is that I want to dive deeper into the psychology side of things, especially like the infrahumanization and human biases. And I also had on my slide, but I'm trying to be good on time, is Slovik, who's an author who talks about like the two system analyses in the brain, the first system dealing with emotion, the second system dealing with rationality, and be able to better bring out how we separate those things. I'll also be working on sampling data and what exactly is permitted and appropriate for my research goals, as I already stated. And then finally, I plan on developing all of these connections, presenting the main ideas I have and covering over summer to show everybody how these things all connect, even though they're completely crazy separate ideas. And that's all. And I just wanted to say thank you, everyone who's been here and been supportive and dealt with me disappearing sometimes for far too long and just being overall insane. But you know, at least we're getting through it together and we have support and we have an amazing family here. And I've told my mom and everyone in my family time and time again, I have never felt so supported in my college career until I joined the McNair. So I just want to say I thank you guys and really appreciate you guys more than you know. If anyone has any questions or comments, let me know.
Thank you so much, Alexie.
Of course. Thank you.
Amazing, amazing. Yes, I echo Emily's comment of, oh, we can't wait to see until you actually get to dig into more primary stuff too. And seeing all these connections, like the galaxy brain does all these different pieces together. Incredible.
Yeah. Thank you. Yeah, I just feel like that's human nature in general, man. You know, it's not just one side of the apple. It's all of it. It's the whole thing, the whole damn thing, the whole reason we're here, right?
Amazing. Okay, I believe now we are going to move over to Emily Duru.
Do you stop the screen sharing?
Okay, can you see / hear me?
Yes, we can.
Okay, cool. So I'm Emily. I'm a senior here at the UO. My major is English with a minor in creative writing. And today my research is an examination on the portrayal of Black girls coming of age in contemporary American literature. My mentor is Dr. Courtney Thorson, and so far it has been really fun. So let's get into it. Okay, so for a little bit of background, I've been an avid reader from a young age. I started off with fantasy books and eventually worked my way over to more like realistic fiction. And I remember being especially enthralled with this series called The #1 Ladies Detective Agency. And for those who are unfamiliar, it's about an African woman who runs a detective agency in Botswana. And as a kid, I just loved these books. Like, it was the representation I was always searching for. But there was also just some language and descriptions in the book that of the Black women in the book that just kind of rubbed me the wrong way. And I really couldn't figure out why for a bit. And then because I grew up in the age of Google, I was able to quickly find out why. And that was because it was written by this man. And while I still cherish those books and they hold a special place in my heart, for me, this got me interested in finding out why these inaccurate portrayal of Black women in contemporary American literature are so harmful. And so, for a little bit of background, in American literature, commonly Black women are portrayed as like the caretaker, the provider, the emotional like counselor, and like really stoic and resilient, but at the same time are not given full emotional like fleshed out emotionally. And so I wanted to explore it even further. And as I researched and found out more about like the portrayal of Black women in literature, I found out a lot of it stems from antebellum mindsets. So dating back to slavery and the idea of like what role a Black woman should play in America. And that has kind of transcended into our literature and is still a prominent part of it today. So as my research developed, I narrowed my focus even further to examine the depictions of Black girls' adolescence, and really how the portrayal of Black girls' adolescence in literature perpetuates the same ingrained ideas that are pretty harmful, that move Black girls from being basically childhood into adulthood without giving them time to mature and truly be children. And rather than focusing on the bad novels, because there are tons of examples of that, I decided to use an author whose portrayals of Black girls and women are more empowering and accurate rather than harmful and archaic. So the author I chose is Paul Marshall. Paul Marshall is a Caribbean American Black female novelist that rose to prominence in the late 50s and early 60s. Her novels, the novels I chose to examine as my primary literature were Brown Girl, Brown Stones, and Daughters. They are about Black girls and women growing up in Brooklyn, New York, under extreme poverty and trying to navigate the world around them, as well as carving out their own space in that world. So these are my research questions. I'm not going to read all of them, but basically they focus around how Black girls' adolescence is being portrayed in Paul Marshall's stories and how we can use those to interpret into our society, and other examples from there that stem from those same ideas. My methodology is mostly lit review and close reading. It's actually just all reading. So a lot of reading never hurt anyone, but for me, I also wanted to make sure that I was drawing out the necessary things. So I chose to compare Paul Marshall's fiction to Black feminist texts and theories from people such as Hazel Carby and other prominent Black feminist theorists. And so the reason why I chose to use those Black feminist texts and theory is because while other theory may be more insightful to things like historical background, intersectionality is a really important part when examining this literature. And for those who are unfamiliar with what intersectionality is, sorry, I live on a really busy street, but intersectionality is when it refers to the interplay between someone's different identities, someone or a group's different identities. So for me, choosing Black feminist text meant that I would be looking at more than just the interplay between like maybe gender in the story, but I'll be looking at gender, sexuality, as well as race. And I thought it would fully flesh out Paul Marshall's fiction a lot better. Yeah. So this is kind of a brief recap. This is me, I cleaned it up a little bit because I used to have sticky notes all over my wall because I would read, I'm reading like 80 books at the same time. So it kind of would get hectic. So I had all these sticky notes on my wall and my roommate said I kind of looked like the picture on the right. So I cleaned it up a little bit. I didn't want to show the rest of my mess because it's just as bad. But basically, there is like reason to this madness. There's an underlying order. So my research could be mainly broken up into two parts. One part focuses on the primary text by Paul Marshall and looking and drawing out examples of like the Black female body, how it's being written, how she writes sexuality, and how she kind of overcomes those stereotypes. And then the other part of my research is looking at the theoretical text and seeing how a Black feminist reading can contextualize and change the meaning of the same words that we're pulling from the Paul Marshall text. So what I've been doing over the summer, well, I can't really say conclusively what my research has shown me because it would take me hours to explain it. But one thing, I'll give an example. One thing my research has shown me at this point is that white femininity is constructed by the degradation of Black femininity. And that is something that is ever present in the way we construct our protagonists and our heroines in our in novels. So for example, in Brown Girl, Brown Stones, I chose the quote from a character in the story called Jyhreh, who is Selena's neighbor. She comments to Selena, who is waiting in the dark hallway for her friend. And she says to Selena, a younger girl, she's 11 in the story, she says, you know what people don't say when they see you always drawn up in some dark place? They don't say you love darkness because your deeds is evil. At first glance, it seems kind of ominous and almost like threatening, that line. But when contextualized through Black feminist theoretical text, Jyhreh is actually warning Selena that while her actions are innocent in reality, like she's just waiting in a hallway for her friend, which is a very juvenile thing, society will not perceive it that way. Society will perceive her, a Black girl in a dark space, as her doing wrong things. And so it's like kind of, so when I applied the theoretical text of Hazel Carby onto it, there's a quote to the right. I'm just going to choose one line, but the dark physical presence of the Black woman who lacks these two essential qualities was constructed in opposition of the heroine. So basically, when you combine those two, it's showing that the outside world doesn't allow Selena and the other Black girls in the story to feel the fragility and the purity that is often associated with childhood, and instead they have to grow up a lot quicker. They have to navigate the world a lot more different than their white counterparts. So the reason why I chose Paul Marshall is because she does a really good job at showing that these characters are not one-dimensional. So oftentimes, like I said at the beginning, when you have a Black girl in a story or a Black woman, she is given just a stoic character. But Paul Marshall allows her characters to feel fatigued. She gives them the full emotional, fully fleshed emotion that they are allowed. They're allowed to feel pain. They are allowed to suffer, like feel their pain. And while they may suffer in the story, they also experience joy. They also are able to speak about their pain rather than kind of suffer through it in silence. And so, yeah, so far I've been noticing a lot of that and kind of drawing those relations between my theoretical text and the fiction from Paul Marshall. So the significance of my research is because I think it's now time, because I have to sit through so many literature classes, and we talk about the Harlem Renaissance, and we talk about all these kind of like older stories. And I think it's time we start to address outdated stereotypes that kind of have plagued English literature for this long. And they've kind of also become ingrained and accepted as fact, almost like the idea that like Black women don't feel as much pain. So in reality, it's actually harmful to Black women, and it's stripping us of our femininity. And I also think that my research will help contextualize the unique struggles of Black girls in womanhood as they enter womanhood, and how we can better give compassion and innocence rather than to say like you're moving too fast and move them straight into adulthood. Yeah, so questions I still have and I still need to go over with my mentor is how can I make my research more intersectional? So a big issue I came across this summer was that I was trying to boil down Black girls and Black womanhood into one shared experience, but that is excluding people who, it's actually a wide diaspora. So like it's excluding trans Black folk, non-binary Black folk, as well as like queer women, Black women. So I'm always trying to see how I can make my research more intersectional and making sure that I'm not ignoring or using a heteronormative mindset. Another thing is why that my research mentor asked me, and it's pretty much melted my brain because that's all I can think about, but it's why do we choose fiction to get as like an example of these things? Because why can't I use like a news report or a nonfiction essay? Like why can't I use actual word of mouth? Why is it important that I go straight to fiction and draw out these examples of misrepresentation from fiction? And that's something that I'm still trying to answer, and I hope to answer by the time I am done with my thesis. Yeah, where I go from here, it's just a lot more reading. I have so many things to read, and I also wanted to read The Underground Railroad, which is a critically acclaimed book. Like Obama put it on his like top books thing, and I'm gonna say it's a bad book because I think it has a lot of tired ideas of Black womanhood that are still being parroted today. Like it's a new, more recent contemporary book. So it's showing that Paul Marshall didn't solve these issues, but she's still a blueprint that we can use even today. Like we still need to look at that for representation. So yeah, that's it.
Thank you so much, Emily. Yay, we might, we will probably trudge onwards, but please, if you have questions or comments, please put them in the chat so Emily can still look at them and respond to them if any questions pop up. And now we are going to shift over to Bailey.
All right. Okay. It is not letting me share my screen right now. But I can just talk, let's see. Did it share?
There we go.
Okay, so my name is Bailey Adams, and I study planning, public policy, and management, and I minor in entrepreneurship. My research is centered around understanding Black Americans' perception of the police. Leading on to that, my research question is, how do Black Americans perceive the police? And I decided to research this because ever since high school, I have been a part of the fight against police brutality and also just the killings of innocent Black Americans. And I noticed that eventually it started to take a toll on my mental health personally. And I had to eventually just take like a 6-month break from that, along with I noticed that I started to react different in certain situations because I had just seen so much of the killings like on social media, within my community, hearing about it. So I just noticed that it started to take a toll. Then recently, after the protest of Breonna Taylor and George Floyd, it led me to want to understand more about what the aftermath is, especially in the Black community. Mental health isn't really talked about, or just our feelings in general aren't really talked about. So I want to know, what do these events, how do these events change people's perceptions in regards to how they interact with police and their attitudes towards them? So my methodology includes doing surveys and video interviews. With the surveys, I'll be focusing on using demographics from like gender, education, or political standing to basically see if there's any change within those demographics, and the questions will be asked as far as how would you describe your views of the police? In what situation would you call the police? How have your experience with the police shaped your view of the police? And I don't imagine it changing too much, especially in regards to people's like education level or income level. But I am interested to see if it does change within people's gender or their political standing. So that'll be interesting to see. And then I'm also doing the video interviews to see if they support the data, and also just to further hear about people's experiences and to tell a complete story. I'm thinking about limiting the data collected to people that range from 18 to 26, because this is kind of my age range, and I'll be able to relate and understand what they're talking about, especially for the video interviews. If they bring up a situation like Trayvon Martin, I'll be able to kind of understand what their thought process is and everything, because I was alive during that time and I understand what was going on. And so I'm planning on doing surveys and using my friends and their friends and other student organizations here on campus to connect with people, and just using that as a way to as a broad net to figure out, to get participants. So during the summer, I have just been working on my literature review and developing questions for my survey. As far as finding other literature, I have not found anything that connects to what I'm doing. It's more so everything explaining like why there are these protests going on, and there's a lot of evidence saying there is discrimination within the police force, but nothing that is like after that, nothing that says like what are the perceptions of Black people after these things have gone on. So I'm still trying to find more. I'm still trying to leave it open to see if I can find something like that, but I haven't come close to anything. And then I will be starting the IRB process pretty soon. That's probably like the next thing I'm going to be doing because I've done a lot of research and I'm kind of tired of it, so I'm ready to start. And then after I get approved, I will be collecting the data, and that is it. Thank you.
Thank you, Bailey. Are there any questions or comments for Bailey before we trudge onwards?
I just really quickly, Bailey, I just said something about the video interviews, but I'm curious of how you conduct those. So maybe we can connect, because I think people's stories definitely need to be heard, and specifically seen. It humanizes people and helps them connect. So I think that's such an important component. So good job. It's awesome.
Thank you. And I'm thinking about just doing Zoom because of COVID and everything. So yeah.
Wonderful. Well, if there are other comments or questions, please feel free to put them into the chat. But now we're going to shift over to Desi.
Wonderful. Hello. Let me get my screen up. Share. Alrighty. Hello everyone. My name is Desi Casanova. I'm a major in psychology. So some of my research interests fall under drug addiction, psychopathology, as well as disability studies for a little bit of background on that. This research, though, in particular, I'm looking to investigate the possible correlation between adolescent drug abuse and deficits in memory recall. So this research in particular, I'm investigating the hypothesis that adolescent drug abuse is positively correlated with deficits in memory recall in adulthood. So I'm looking at the long-term cognitive deficits influenced by adolescent drug addiction, and specifically what the adolescent is doing to their brain during this really important development in their life. So through this research, I'm utilizing individual perception. This isn't like a big study the way that I wanted it to be originally. I've definitely had to narrow the study down quite a bit. So now I'm just utilizing a survey and seeing how people feel about their own memory after having experienced some sort of drug addiction during their adolescence, and now reflecting on that and how their memory might have been affected by those by their drug addiction. So this is really to build on current research on these various deficits. Unfortunately, there's not a lot of research being done specifically with adolescents because it's really hard to do. But it's important because we're looking at the damage being done and how that's being carried on into adulthood. So that's really what this research is looking at, is seeing where these deficits lie. So for a little bit of definition, what do I mean by memory recall? Well, memory recall is the process of successfully being able to retrieve information you had previously learned, experienced, or obtained, you know, that's relevant to the situation, and then be able to reiterate that information to other people. And then defining drug abuse. So I'm defining drug abuse as the compulsive act to use either a single psychoactive drug or multiple psychoactive drugs that are impacting life in a negative manner. And I'm specifically looking at drug addiction between the ages of roughly 12 and 18, I would say, are my goal factors. So if a participant in my study, if they experienced, you know, some sort of addiction, and this can be with any sort of psychoactive drug. I'm not, you know, specifically looking at one or another. You know, it can be as small as even cigarettes to as big as, you know, heroin, methamphetamine, anything that they had the compulsion to keep using and it negatively impacted their life during that time. So those are just some definitions for you before I continue. Why do we care? Why do we care about this? So we care about this because it is so relevant in today's time. You know, so many young individuals are suffering from drug addiction. And if we can look at the cognitive deficits that are being influenced by drug addiction in this time, maybe we can utilize this to develop new counseling strategies. Maybe we can utilize this to develop new new treatments that need to be done. And really, I'm utilizing memory recall as the basis of this study, so we can really just start the push for this research, start the way that we can look at different areas of the brain and determine how drugs are affecting someone long-term in their life. So it's important. How are children damaging themselves, and how can we help them later on in life? So what's been done before? Unfortunately, there's not a lot of research looking specifically at drug addiction and memory recall, right? So there's a lot of short-term studies being done on like very short-term cause and effect kind of thing, but there's not a lot of longitudinal studies that are happening as far as following the individual for a long period of time and then seeing what the correlation might be. So there's really what I've been doing is I've been piecing these two subjects together. So I'm looking a lot at adolescent drug addiction, the short-term cognitive deficits that are happening due to their addiction, and then I'm also looking at memory recall and how it's stored in our brain, and then how drugs affect these pieces of our brain, right? So a lot of what I've been doing is piecing these two subjects together because they've never been studied in the same domain before. So it's been a lot to look at and start to bring these pieces together. But really, you know, it's time that we start to look at the brain as something that can become injured, right? So a lot of it is treating the addict when we should be treating the brain. And that's why this research is so important in how we look at the damage that's being done and why we look at it. So specifically for me, the methods that I'll be using for this research is I'm just doing an open online survey. So anybody can take this survey. I'm hoping to gather many, many participants to really have a broad view of the individual perception on memory recall. The length will be roughly 30, 35 questions. I don't want it to be too terribly long. I want my participants to be engaged. And my population for this is definitely individuals 18 years or older. So like I said, anyone will be able to access this research. But if I have participants that are under the age of 18 and they will be asked their age, their answers will not be recorded in my official data, because it's really important that I'm just looking at adults and just recording their answers. My participants can remain totally anonymous if they'd like. They don't have to add their name whatsoever, or they can use an alias if they want to, or by choice they can disclose their name, but that portion will be completely up to them whether they choose to disclose that with me or not, because there will be some sensitive information that I'm asking throughout the survey. And again, anyone can take the survey. I really hope to get lots of participants. And there's no requirement for needing to have experienced drug addiction, right? Like, I'm not specifically targeting those people. Like, I won't just be sharing it with people in, you know, drug groups or anything like that, you know, so it's not required that you have experienced some sort of drug addiction to participate. In fact, you're super encouraged to continue to participate in this if you haven't, so that way I really get a broad understanding of those who did experience and those who didn't experience drug addiction. So those things are really important, and I hope to get a lot of participants using this really open, free format. So some of the progress that I've made so far this summer is a lot of it has been my literature review. As I stated before, I've really been piecing together these two very different domains, you know, from looking a lot at the brain and the hippocampus and the amygdala and how those things store our memory, and then how those things affect the way we reflect on our memory. You know, the hippocampus is where we store new memories and where new memories are created, but then the amygdala is our storage, and then it also is how we reflect on those memories. So if you're having a really negatively impacted memory, the amygdala is going to really be the storage for how that is perceived later on. And then the same thing with drug addiction. You know, these things are causing or are related to more like how we're end up perceiving things later on. So I've been digging deep into this literature and really putting these pieces together, which has been quite the feat for me. And then I've also been working on IRB approval, working and very close to having all of my materials that I need for that to submit, and start hopefully open up my research here. My goal is to open things probably like the 1st of November. That's like, I think that's a pretty obtainable goal, is to get my survey up and out to the world for people to begin participation. So IRB approval is being worked on currently, and then building my survey, of course. And I've been talking to other researchers like Sarah Dubrow and Ruth Ellingson to kind of develop that in a way that is engaging, but is also going to answer the questions that I need answered. And then of course, outreach, talking about my research and, you know, really bringing people into this to get as many participants as I possibly can, because that's super important in this. I need many participants of all stages of their life. And so I'm doing a lot of outreach. I'm talking to a lot of people. I'm talking to friends and family and faculty. So that's been a really big portion of this. And next steps. So like I said, I am a senior this year, so I'm applying to the Oregon Health and Science University. I'm applying to their doctoral program in clinical psychology. And then there, which is where I can continue the development of this research and hopefully do what nobody's done before, and that's create a longitudinal study where I'm following individuals over the course of several years and really looking at what's happening to the brain as they continue to develop after having experienced drug addiction. So that's really where I want this research to go. And in this, I'll be specializing a lot in psychopathology, disability counseling, drug addiction studies. You know, those are the big overarching goals for me in this research. And I'm really excited to see where it goes. And that's it. So thank you everyone for listening in. I'm really excited to continue to develop this research. I'm really excited to continue to work with McNair. You guys have been a huge support, and thank you.
Hey, thank you so much, Desi. Amazing, amazing. There are some comments and such being put into the chat. So I encourage you, as always, as I have for the past now 7 presentations, please feel free to put things in there because that is a great landing spot. And I want to move us next to Alyssa.
Perfect. Okay, can everyone hear me? Perfect. Hello, my name is Alyssa Fuentes, and the project I've been working on this summer is wide field imaging of visual cortex in environmentally enriched mice. I've been able to work on this project under my mentor, Denise Piscopo of the Neal Research Lab at the Institute of Neuroscience here at University of Oregon. I will begin this presentation with just a general overview of the visual pathway, followed by detailing some environmental enrichment studies as they relate to vision. I will then present my question and hypothesis. Following, I will detail the chosen approach of wide field imaging while also noting some of the types of information we can gather from this approach. I will go over my summer progress as well as future directions for the coming fall semester. And then finally, I would like to end with just some acknowledgements. So an image enters the eye as light, where it will reach photoreceptors of the retina. And these photoreceptors are responsible for converting this light stimulus into an electrical signal. And it's the passing of electrical signals, which is the mode by which signals are communicated within the brain. And thus, these electrical signals are passed along the visual pathway. So it starts in the retina. And something notable about the retina is that, and this is common throughout all of these landmarks, so retina, LGN, and primary visual cortex, and HVAs, but it contains a mirror image representation of the visual field. And we call this a retinotopic map. So electrical signals are sent to LGN, which is located in the thalamus of the brain. And this retinotopic map is maintained. Some lower order processing occurs at this level, but for the most part, LGN serves as a relay station and sends information to primary visual cortex, which is known as B1. This is the first location of higher order processing where linear boundaries of objects are extrapolated because the neurons in this location are specialized to respond best to lines. V1 is in close communication with higher order visual areas known as HVAs, and it's through looping communication of these areas that allow for complex processing like perception. Environmental enrichment has a profound impact on brain development, function, and plasticity. In a study, in a collection of studies by Hubel and Wiesel, they determined several key concepts in vision. However, I want to detail 3 of them. First, they determined that at birth, there is some level of organization within the visual pathway, but they found that with deprivation, those connections can actually degenerate. But then also experience instead can strengthen those neural connections. So this kind of nature-nurture concept. And then in a study that looks more directly at environmental enrichment, Griptu and colleagues found that environmental enrichment can actually preserve, strengthen, and restore plasticity in adult mice. Now I'm only noting a few findings in a few studies, but several labs have demonstrated that environmental enrichment can alter neural function, inhibitory circuitry, gene expression, neurotrophic factors, and other physiological properties. This brings me to my topic of interest, which is, does environmental enrichment affect neural activity in primary visual cortex and higher order visual areas? This is significant because although the benefits of environmental enrichment has been established, the impact of such an environment on V1 and HVAs in whole population recordings has never been quantified. And this is essential because the majority of vision experiments, the mice are housed in standard conditions. These standard houses could potentially be inducing a visual deprivation and thus affect the efficacy of results for vision studies. So when presented with this question, we hypothesized that an environment enriched with social, physical, and visual stimuli increases neural activity across V1 and HVAs in the adult mouse. For our experiment, we have mice that are housed both in an enriched environment and standard housing conditions. We define our enriched environment by providing social stimuli where mice are housed in cohorts for socialization. We also provide physical stimulation, which consists of a double layer maze and running wheel. And then we also provide visual stimulation where mice are given an iPad. Where mice are given an iPad. Both cages are provided materials for nesting, water, food, and a shelter. The imaging method we chose was that of wide field imaging. Our mice have a brain that essentially glows when cells are active, and this fluorescent indicator is incredibly sensitive, and it allows us to image the brain through the skull. So here's a schematic of the wide field imaging apparatus that we use. And what you can see is there's a mouse head fixed onto a spherical treadmill so that it can freely move during experiments. And then above it is positioned a custom macroscope that rapidly images the brain while we present various stimuli. One stimulus that we use is what's called natural images, and it's actually a series of images. So in this example, we have a cactus right here. These images include landscapes, animals, and plants, and were chosen because they present a complex stimulus. The other three stimuli consist of contrast gratings, as pictured here. And they appear either as a scrolling bar along the X or Y axis, and you'll see this in just a moment, or they can present as a circle that momentarily appears and then disappears at different locations on the stimulus monitor. And we chose these stimuli because they are optimized to activate as many photoreceptors as possible and thus maximize neuronal excitation. The scrolling bar stimuli are incredibly important because it allows us to generate these retinotopic maps, which are essential because this is how we can actually define the boundaries of V1 and HVAs, which are areas of interest. I am including a movie right here. So in the top left corner, what you see is this scrolling bar stimulus. And this is an image of a mouse brain. So we're actually looking down on a brain. And what you can see is that whenever the stimulus is scrolling across the screen, you see a simultaneous fluorescent bar scrolling across the brain. And this is indicative of neuronal excitement. So for this summer, I finished up some wide field imaging. I also focused on a literature review to gain a more comprehensive understanding of the visual system and my project. I also sought to learn Python 3s. Our analysis isn't done in Python, it uses MATLAB, but Python 3 is more widely used, so I thought it would be more advantageous for me to use that or to learn that. And then I'm going to be using some of the same principles that I learned from Python and apply them to MATLAB so that I can understand the code. And then finally, I assisted in separate environmental enrichment behavioral experiments that are not in the scope of my McNair research project, but also gave me some good lab experience. And looking towards the future, I have some new enrichment cohorts coming in, which I'm very excited to image. And then I'm going to be diving in on analysis to see what our results actually are saying. And then I am going to continue to assist in some separate environmental enrichment behavioral experiments for some more experience. And I would like to thank my mentor, Denise Piscopo, for helping me every single step of the way. Wendy and Vanessa are undergraduates that I work closely with on the enrichment projects. Kevin, Keegan, and Denise, thank you so much for helping all things McNair. And then the Neil Lab is truly an amazing lab that I am blessed to be a part of. Thank you.
Thank you so much, Alyssa. Because we are so close, I might, again, comments, questions, please absolutely put them into the chat. All sorts of science things. It's been a wild morning. We have one more presentation, wonderfully, excitingly, stressfully, perhaps, and I'll turn things over to Nicole.
Okay. Sorry, 1 second. Just trying to get this. Okay, sorry. Okay. All right, usually I just hit F5 to start my slideshow, but it's not working. Oh, here we go. Okay. All right, so hi, I'm Nicole Wales. I'm in the Core Win lab at the U of O's physics department. So we study glass because it's one of the great unsolved mysteries of physics. This is because glass is molecularly disordered like water, but it's rigid like a crystal. So you can blow glass, right? But you won't really see anyone blowing water. And so we don't really understand why glass has the properties that it has. So to contribute to the overall understanding of this, even in just my own small way, I've been studying the correlations between simulated glass particle configurations, which are also called arrangements or packings. And specifically, I'm looking at those that neighbor each other in phase space. So that's a mouthful, but I'll explain what that means a little bit better in a minute here. So specifically, I've been looking at three-dimensional jammed athermal systems. So an athermal system is just one that's at 0 temperature. And it seems silly to study something like that, but the reason we do is because it's a useful way to understand a system's behavior as a result of its geometry without worrying about what the temperature is doing. So when you think of like a jammed particle system, you can imagine putting so many marbles into a box that when you shake it, you don't hear them all rolling around. So that's just to get a few definitions out of the way. So my project requires that I compare the configurations of nearby energy wells on the energy landscape of a glass particle system. So an energy landscape is just a map that associates all the configurations of a system to different energies. And energy wells, which are also called minima, correlate to more stable low energy system configurations. Meanwhile, peaks or barriers correspond to unstable high energy configurations. And here we have two neighboring minima labeled well A and well B, shown with their associated configurations. And then I have them, so I have the white and the pink outlined configurations. Oh, and just keep in mind, these diagrams,
They only show 5 particles just for simplicity, but you can imagine that the rest of the black space in these boxes is filled with other particles that form a jammed system. Okay, so when you're comparing the associated configurations of 2 neighboring minima, like A and B, if most of the particle positions are different, then a global deformation is responsible for their differences. So here you can see the white and dashed pink configurations do not perfectly overlap. So all particles have changed position going from one configuration to another. That means our configuration A underwent a global deformation to become configuration B, or vice versa. So if few particles change positions when going from one configuration to another, then we have a local deformation. So here only a single particle, the top one, has a different position when you compare the 2 configurations. So these definitions lead me to my research topic. If I have a jammed 3-dimensional and athermal particle system, do the configurations associated with nearby energy wells differ by global or local deformations? So you may be wondering why this even matters. Well, understanding configurational differences is significant because it will tell us if the global deformations that are predicted by the mean field theory in the limit of infinite dimensions holds in lower dimensions, such as the 3-dimensional systems I'm looking at. So if the prediction holds, a newly predicted phase of glass, which is called the Gardner phase, may be universal amongst all amorphous materials in general. So that would suggest we can apply what we learn about glass to a wider variety of materials. And this kind of insight may lead to the development of stronger new materials, which is pretty cool, but honestly kind of far off in the future. But if our nearby configurations are correlated by local deformations, then we need to understand why the mean field theory's prediction fails in lower dimensions. So my method. So my initial approach to answer the research question was to find the Cartesian distances between corresponding particles of nearby minima configurations. Use these distances to calculate the inverse participation ratios between minima, and use the sum of particle distances to define the distance between the 2 overall configurations. Then I would just plot the IPRs, or inverse participation ratios, against those configurational distances for analysis. And I'll talk more about the inverse participation ratio soon, but just know that it's a good indicator of how many particles move going from one configuration to another. So it makes it a great tool for deciding if I have a local or a global deformation between 2 configurations. And so some assumptions that we make in lab. First, we assume that rattlers can be ignored, and those are just unstable particles that make no contacts to other system particles. So it's thought we can remove them with no major consequences, but we'll see. So if the red particles in this diagram are rattlers, then neither of those particles, in this case 3 and 5, are going to be included in our calculations of distance between the particles of the 2 configurations. So we would just delete those completely and look at just the ones that have that are stable in both systems. Second, it's assumed that periodic boundary conditions are not a problem for the calculations. So periodic boundary conditions are used to approximate a large or infinite system. Outlined in the purple unit cell, we have this repeated pattern. And so the particles that are cut off at the top and bottom borders are the same particle. And then the particles cut off on the left and right borders are also the same particles. So you can kind of think of it as like mapping this 2-dimensional example onto a torus, or like a donut shape, if that makes sense. And then we have global drift. So to get the Cartesian displacement between neighboring minima configurations, we have to account for this. It occurs when the unit cell has shifted, which can be misleading and make it appear as though a global deformation occurred between configurations. So in reality, there is no difference between the configurations in the purple and red bordered unit cells here. All right, so to get our minima distances, we just subtract the XYZ positions of the corresponding particles of two configurations A and B to get these cyan displacement vectors. Then particle 5, if you notice, it didn't move, so it's just labeled with the 0 vector, which is just given with a 0 and then an arrow above it. Then we get the mean displacement of particles in each direction, and the blue vectors here show the mean displacement of configuration B particles from those of A. So we subtract those mean displacement vectors from the configuration B particles to yield a new configuration, which is just shown here in purple, and that's going to be our new configuration for our B particle. And then finally, we can use the Pythagorean theorem to get the distances between the configuration A and new configuration B particle positions. So the diagram shows these distances and as vectors in red, orange, yellow, green, and blue. And then the distance between configurations A and B is then just the sum of the particle distances. And that's shown as the length from the red tail to the blue arrowhead at the bottom of the diagram there. Okay, then we use the particle distances to get the inverse participation ratio, which I'll just call IPR. And it tells us how many particles have moved going from one configuration to another. So for example, in a system of n particles, an IPR of 1 just means that one particle moved, and that's a local deformation. An IPR of 1 over n means that all n particles moved, meaning a global deformation. So it's taken me all summer to learn Python and write scripts that are efficient enough to run IPR and configuration distance calculations on massive sets of data. So the IPRs are then plotted on the y-axis, and the configuration distances were plotted on the x-axis. And these are given as log-log plots. And I have, just as an example, just 4 plots here for the system sizes that I analyzed. So those sizes are 64 particles, 256, 1024, and 4096. And let's see. So every data point represents the comparison of 2 minima pairings with a total of 500 minima for each system. So we're talking about a lot of calculations, a lot of comparisons. My initial plots were created from data where I ignored the rattlers, like I said before, and those are shown as pink dots. And then because of the ambiguity that comes with rattlers, I also calculated the IPRs and configuration distances for the same systems with the rattlers included, and so those are the blue triangles. I've only processed a little bit of data so far, but what I've seen, it looks like global deformations may dominate and being responsible for those configuration differences. So if you notice, there aren't really a lot of data points that approach 10 to the 0 on the y-axis. So that's just 1. Or it corresponds to an IPR of 1, which would suggest a local deformation. So you'll notice a much higher concentration of data points are in these much lower y values, which points to a global deformation for a lot of these. And so my next step will be calculating the IPR in contact vector space, which is based on the contact vector network. So if you're given 2 configurations A and B, the stable contact vector network would be shown in purple and cyan, respectively. So it's just where they contact each other, basically. So from the displacements between the corresponding configuration A and B contact vectors between each particles I and J, I can calculate the IPR in contact vector space. So that is my next step. So once that's written into my script, I can finally process all of my data. All the data previously taken in my lab, and then I can start running my own new simulations based on what I learned from that. And that should be plenty to answer my research question, or at least point me to relevant new questions. And yeah, I would like to thank the McNair Scholar Program and Core and Lab and the NSF and the Simons Foundation. All right, and that is that. Hopefully that was understandable.
Thank you so much, Nicole.
Sure.
So I think at this point, because somehow we finished before noon, which I am so proud of us for so many reasons, because that was a whole lot of information that you all threw at everyone, and in the best possible way. It's not all that, but wanted to see if there are any questions for Nicole, or because we have a little time left, and because I always think it is cathartic and fun, and we have some fun special guests, if there are any comments or encouragements or whatever from anyone who may or may not, or who is here and has been able to help bear witness to your academia-ness.
Hi, everybody. Hey, I wanted to go back to Emily Boyce, if Emily is still around. So now on the study for the prenatal part of the skeletals, how would that be done, and how have you been able to dig into that piece of it? Because I'm going to be a grandmother soon, and I'm really fascinated about the prenatal piece of that.
Oh, I think you might be talking about mine.
Yeah.
Yeah,
Emily
so,
Boyce, I'm sorry.
yeah, so the research that's already here is that when, I guess you can say they're in the fetus.
I'm sorry, I've got my names wrong.
It's okay.
Go ahead.
But certain bones form during certain parts of a female's trimester. So there's actually this is super cool visual that I had, but I couldn't put it in there because it wasn't really pertinent to what I was doing. But I can send you the link for it. It is adorable. It goes through the whole transition of when these certain bones form, and what they look like, and how itty bitty they are. But in terms of the skull, actually, I can tell you more about an infant skull is that their sutures aren't fused yet. So that's why when we're babies, we have that soft spot on the top of our head. So it's just a little open space. So please, when you're holding the baby, be very careful. Their bones are very fragile and very, not brittle, but they can break easily. But there are studies showing that during the process, there different bones form, and it's cool to see it form too. But they are doing certain research to see is it a genetic process of why these certain bones form before other ones, or are the long bones going before the short bones, or the itty bitty ear ossicles forming in their order. So I can totally send you those, Denise.
Oh, that would be awesome. Yeah, I've seen a couple of the ultrasounds, and she's just 21 weeks right now. So first grandchild, you know how it is. It's oh my God. So I'm sorry about the name mix-up, you guys. I had it written down properly, but it didn't come out right. So, but thank you, every single one of you guys. You're so inspirational. I look forward to knowing you guys a lot more during this remaining of this year. But I'm so inspired by all of you guys, and thanks for everybody that came today.
It has been very fun for me to try and a version of Drill Sergeant all of you this summer, as I have pestered you with emails and check-ins and assignments in class, and asking you so many questions about your projects and trying to understand them as we have gone through this journey together. And so I feel like I alone have known the most bits about most of your projects until now, because I've been communicating with all of you so regularly. But being able to hear you all be professional and versions of experts, even though I'm sure it doesn't quite feel like that yet, because you're still going through stuff, but you are the ones that hold so much of this knowledge. And that is because you are so immersed in it. That is a unique experience in the world right now. And so this, I think, is the beginning of kind of showing how formative and exciting and developing these experiences are for all of you as you continue in research and continue in graduate study, as this is a version of what you'll be doing in the future for many of you. It's been fun for me to hear about all of the learning moments of, oh, I didn't realize I could do it like this. Oh, I didn't realize I need to be as rigorous or as annoying to my PI or mentor to get these answers. And so being able to hear about process pieces as well as content discipline pieces has been so much fun for me, because it's been a way for me to also just learn more about departments, all the different departments and mentors you're working with, and kind of now I have fun party tricks to share with all of my cool friends who are in one specific discipline. I'm like, well, I know some things about some other things because of these super cool students I'm working with. So I want to thank you all for the privilege of being able to support you in this work. And I cannot wait to continue forcing you to do things this fall as you continue developing these projects and figuring out what the next step looks like for your research and for your careers.
Hi, and I'd just like to add in, my name is Barbara. I was very inspired by all of you today. I learned a lot today, and I will definitely be using you all as examples and inspiration for not only other UO students, but I work primarily with high school students. And so I'm very excited to just to use you all as examples of what you can do when you come to college. So thank you all for sharing all of this, and I wish you success with your continued research.
Hi, everyone. This is Christabel. I completely second what Ms. Barbara just said. And I, as alum, am just so, I'm relieved with you, I think, because I know that this works in progress part is a big hurdle, and there's one more other kind of hurdle to happen to sort of identify as a McNair scholar. And I am so excited to be able to have been here today to watch you all. And Jyhreh's mom, are you still on? Because yes, Ms. Johnson, I have something to say to you. I'm so happy we have parents representing, and I have to tell you, I have such fond memories of talking with Jyhreh freshman year. And to see her growth and to be able to sit here and witness her just amazing development and how well she articulates her research, and it's just awesome. And we're so happy that you were able to join this and to also, again, witness her development. And she literally made me cry today. But I am just so excited for those of you who I've met already. Great job. For those of you who I haven't met in person, you should all be so proud of yourselves. You just killed it. And what an honor. And I hope you take a moment to sit back after this Zoom call, maybe after today, and soak it up and just relax and just treat yourself to something and enjoy, where you're at and how far you've come. It's just awesome.
And just, I can't really build on that, but Barbara, thank you so much for spending the morning with us, and Jyhreh's mom, it's awesome to have you here. And what I'm hoping is we can all reconvene on May 27th, 2021 for the big undergraduate research symposium. And then we'll be able to celebrate the conclusion of all of this. So no, this is so cool. And I love this milestone in kind of late summer, early fall, before we kind of make the turn into fall term. And again, as everyone said, to kind of take a breath and celebrate. And it is such a privilege to get to with all of you. And one of the things I think that was really a great opportunity is, as you're learning to present in Zoom, one of the benefits of the McNair program usually is to send you to go to conferences to present your research physically. And until we can fully resume that, we're going to be really looking for opportunities for you to continue to present your research at regional conferences, national conferences, but for the near future, at least, it's going to continue to be in a Zoom platform. And as you all demonstrated today, you are awesome at presenting at Zoom, and the PowerPoint presentations So congratulations. And that's the nice thing. Now that you've done all this work, you can easily do it again and present to other audiences. And so that's something we'll be looking at in the fall, is other chances for you to present your research. So thank you all. It was such a privilege to be here this morning. And I want to thank all of our guests that joined us as well. But great work, everyone. This was awesome.
Great job, everybody. Great job.
Well, so you have all survived. You're all still here. You're all smiling somehow after the pain and torture we just forced you through as part of the McNair program. So with that, I believe we can adjourn for the day. Thank you all so much for your time and energy and witnessing and energy and all that. And we will, as always, continue to be in touch about next steps and what next week looks like. And I know that we will continue conversations about this research and what you want to do next and all the super cool things you've already done and the things that are still to come. So with that, have a good rest of your Tuesday, everyone.
Congratulations, everyone.